I will turn it over to Jim Patton to introduce today's speaker.
You have to get up. I'm not gonna try and walk over the cord. So it's a pleasure to welcome back a tag team match. Zelditch and Don Swiderski, husband and wife team from the University of Michigan. They both trace their academic lineage back to this institution, although Don wasn't actually a student here at one time, and I'll say a little bit more about Mimi's role in her early days here. I thought that we were going to have a tag team match the presentation, but out of family harmony issues, they decided to flip a coin, and Mimi either lost or won. Look at it, she gave the presentation today. So these two together, most people, maybe or most of you in the room know that they've been coming back here for at least 15 more years, close to 20 years, to give a geometric morphometrics workshop every summer to the students and faculty and staff of Berkeley and other universities around here. So they're well known to this place. They're also well known in the community writ large of morphometrics and morphological evolution, because they generated the textbook, the Bible of geometric morphometrics. It's been what is now in its second edition, something like that. Geometric morphometrics for biologists.
Chuck got us to do
There's
it.
a copy of it back there. Anyway, they're big names in this field. They helped establish the field for looking at the evolution and diversification of biological shape as opposed to just size kinds of parameters. So we'll hear a lot of that in Mimi's talk. I just wanted to say a couple of things about Mimi specifically, because she traces her lineage back to MVZ from the mid-70s, when she was initially enrolled on campus and as a student in the philosophy and history of science. Came down here for some reason and took David Wake's evolution course, and like David and his course, for a lot of people, changed their trajectories. It changed Mimi's from the philosophy and history of science into being a real scientist. She was actually a curatorial assistant in the herp lab in the early days, and then she went on to the to Michigan State University to work initially with Jim Edwards, who was one of Dave's students, and then when Jim left Michigan State to go to NSF as a program officer and then as a permanent employee at NSF, she transferred over to Don Straney, who was another student out of the MPC. He was one of my students. And Don got his degree with Don as Straney as well. So that's why I say they both trace their academic lineage back to this place. So these two together, as I said, have kind of centered the field of geometric morphometrics in a very broad context. So I had the pleasure of writing a letter of recommendation for Mimi for promotion last fall, because I'm just going to highlight a couple of things in some of what I said in that letter, beyond what I've already said. So they address fundamental and wide-ranging issues in lineage diversification, including broad topics like developmental ontogeny, developmental integration, evolutionary lability and integration, modularity, heterochrony, disparity, canalization, plasticity, and adaptive and non-adaptive radiation. I went on to say this is an expansive list, one that emphasizes in my mind that she, and I should say both of them together, are not shy about not only tackling important topics, but taking a leadership role in the resolution of those topics. And so it's a real pleasure to have both of them here, even though Mimi is the one who's going to give the talk, and so it's a particular pleasure to have you back home.
It's routine for a speaker to say thank you for the introduction, but to still be blushing probably 10 minutes into it is an unusual experience. It's always good to be home, and I'm hoping that I'm going to figure out how to manage this technology when I'm not used to advancing the slides. Well, I've got the mess. So the reason this talk is on adaptive and non-adaptive radiation of squirrels is because I needed a title in advance, and I figured I would find something to say that could broadly fit in this topic. So it's going to have something to do with adaptive and non-adaptive radiations of squirrels, among other things. So one of the questions that evolutionary biologists and ecologists ask is what accounts for exceptional diversity? Either diversity of lineages, diversity of regions, and we'll also talk about diversity of morphologies. Well, one explanation is God has an inordinate fondness for beetles. And it's obvious God has an inordinate fondness for squirrels. But God's inordinate fondness for beetles can only be part of the story. So what's the rest of the story? This slide actually took more time to work on than any of the others. And it's not like I think it's complete, but at least it's a start at what's the rest of the story. Time, rates and modes of diversification and divergence, ecology, and geography. So time is obviously important in that all else being equal, older lineages, earlier colonized regions will be more diverse and disparate than younger ones. Rates are important in that all else being equal, faster diversifying and diverging lineages will be more diverse and disparate than slower ones. Now, adaptive radiations have been one of the main focus of research for a while now. And it's not, adaptive radiations may not be equivalent to this early burst idea, but they've become virtually mathematically defined by this model of explosive rates of diversification and divergence that decline as ecological or geographic space saturates. And all else being equal then, disparity depends upon the range or the volume of the ecological space, since things stop when that's filled. Non-adaptive radiations, it's not that there's no adaptation, but explosive diversification without morphological divergence. And all else being equal then, disparity depends upon the equilibrium variance around a stationary optimum. Late bursts are almost never talked about. There are, I look for examples to show on the slides, and it's rare to see anyone arguing for a late burst. But rates of diversification and divergence accelerate towards the presence. So, okay, squirrels as a model system. You know, all this stuff about exceptional diversity and radiations might be interesting. What does it have to do with squirrels? Squirrels are not exceptionally diverse. Squirrels in the green are much less diverse than Chryseididae in the brown and Muridae in the orange, even though the lineage of squirrels is much older. Now, we say it's low diversity, somewhere around 285 species. So if you're working on a tiny group, that might sound big. If you're working on rodents, 285 does not sound like a large group. Squirrels are also famously conservative. I could quote people in this room on the subject, but here I've taken quotes from the literature. Compared to Muridia, its members display more constrained adaptations and morphologies. And among living species, morphological differences tend to be subtle, continuous, and quantitative, involving shifts in size and continuous graded proportions. Sciurus, one genus, is even interpreted as a living fossil. And Sciurus was designated a living fossil based on similarity of its postcranial material and mandibular morphology to Douglassaurus jeffersoni, which is from the late Eocene. And it's arguably the ancestral squirrel. So that's the mandible of Douglassaurus jeffersoni. That's the mandible of the species of Sciurus in Michigan. And there's an example of what postcranial morphology can do for squirrels. It's a modification of the ankle joint. Now, obviously, some lineages are exceptionally uniform morphologically. I've seen more variation than this intraspecifically in some groups or some species. This is 9 species of chipmunks. But squirrels do not all look alike. This is my favorite slide in the talk, so I'll hold a minute so you can appreciate it. Nor do their jaws. I have noticed that one and that one. They're alphabetically one right after the other in all of my files, like photographs of the data. So it's not going from the sublime to the ridiculous, it's the reverse. That is sublime. And they've also diverged along many niche dimensions. Macrohabitat, they live in varieties of different environments since they're distributed worldwide. Deserts, mountains, paleo rainforests, boreal forests. Differ in micro or meso habitat, whether they prefer rivers' edges or more open spaces or more crowded forests. In space use, Louise Emmons is the one who taught me how to think about niche partitioning in general and in squirrels in particular. And her work on how 9 species of protozoarian squirrels can coexist in Gabon. One of the points is vertical height. We don't have the canopy of an African rainforest here. Well, in North America, what you more often see is the horizontal spacing, how far from the trees, or how far from various other objects do they prefer to forage. The activity time, locomotory, foraging, locomotion, and escape strategies. Diet, including feeding behavior, the preferred material properties of food, size and hardness, as well as specialized resources, and body size. So here's one dimension of niche partitioning, is just locomotory. And one distinction, arboreal glider. This is not usually classified as an arboreal species, but obviously it can climb the trees. Most importantly, you can see it climbing down head first. This one gets up there. It cannot climb down head first. So another dimension of niche partitioning is diet. And I would love to have many more pictures of squirrels eating net, what they actually do. But it's sort of hard to get photographs of them while they're eating, unless you're feeding them. But I saw a list of some of their resources. Nuts, including things, let's see, bears. I certainly would not try to eat that, even with a nutcracker. Seeds, pine cones, pine needles, fruits, leaves, fungi, arthropods, grass. And so I have my gratuitous cute pictures of dietary diversity. Peanut butter, corn, pumpkin. We grew pumpkins for them, we grew watermelons for them. We thought the food was originally for us. But we didn't get it, so we got used to finding half of each strawberry pile along the walkway. And they're so not such cute. But it's clear that squirrels can eat a variety of things and will eat what they can get. So what we're trying to do is link evolutionary dynamics, disparity, adaptation, and coexistence. Obviously, it's a small research project. So adaptive radiations are one explanation for high disparity. But in general, we don't know what processes account for disparity. One issue is many studies use models to select which one best accounts for the dynamics of disparity or diversity, but they don't actually measure disparity. So we don't know what processes produce the most disparity in any case. Now, adaptation is presumably at the heart of adaptive radiation, but it does need to be documented. I did take Dave's class. And that is difficult if convergence is rare. Because when we're using phylogenetic comparative methods, what we really need are independent acquisitions of the ecological state and the morphological state. And there's no predictable association between ecology and phenotype across lineages if convergence is rare. Now, coexistence early in the history of the group is thought to be one of the main promoters of adaptive radiation by a burst of niche partitioning. But coexistence does not require that. If species are disparate, they've diverged long before they coexist. So adaptive radiations might not explain exceptional regional patterns of diversity and disparity and widespread lineages, as long as they have diverged and their biogeographic history brings them together after they've diverged. So what I'll start with is on evolutionary dynamics and disparity. Now, in our previous work on looking at whether rates of morphological divergence are related to rates of speciation, we analyzed rates of speciation for the groups that we had. And what we found is, on a background of decelerating rates, we see 3 bursts of acceleration. There's chipmunks that all look alike, early branch of marmotine ground squirrels, and one clade of Meso or South American tree squirrels, for which we had too few species to be able to try to pin down. We had used everything that had been sequenced. Some of the South American tree squirrels have been sequenced since that paper, so we've been redoing the analysis. There are alignment issues, so we just analyzed the tree squirrels. And it's still somewhat ambiguous, but Central and South American radiation. So it's not just entry into South America, from Central to South America. And unlike most groups of rodents, they actually did go to South America 3.5 million years ago. And maybe there's another burst of speciation on this undershelf. So to measure jaw shape disparity, well, to analyze disparity, we're using geometric shape analysis. So we have these discrete anatomical loci, and then, as you can see, that's not much for characterizing something as complex as a curving jaw. And that curvature matters because there are no actual landmarks where muscles insert. So to analyze jaw shape, we're using these landmarks and then semi-landmarks that are just positioned around the curves of the jaw. So what this shows is, first of all, this is a principal components analysis, just colored polygons for lineages, which is showing you where some of these points are. That's sublime, that's ridiculous. That's another oddity, that's one of our miniatures. Some flying squirrels get really weird. That's the phylomorphous space. So what it does is ancestral shapes are inferred and then added to the shapes of the observed species, and do a principal components analysis of the combination of the inferred ancestral shapes and the living species, and then the branches linking ancestors to descendants are drawn on the plot. So the length of the branches are a function of how dissimilar the morphologies are, not how long the time is, although you can get a time axis if you want. Now, working with complex high-dimensional data like shape does present problems. There are 2 coordinates for every one of those points. And nearly all methods for evaluating models of divergence are restricted to univariate data. They've gotten extraordinarily sophisticated. People spend lots of time talking about all the technical details of these methods. There are new ones always being introduced, and they work only for body size. Only body size can be adequately measured by a single number. All the rest of morphology can't be reduced to a number. Now, we have more variables than we have. We've measured 186 species. We have 196 coordinates. We obviously are not going to do anything that requires estimating parameters. And shape cannot be reduced to a collection of one-dimensional variables. And that's just actually from the definition of shape. Even if you think if you want to be able to do it, or if you're philosophically opposed to it, it's mathematically not possible. What we have done, and it may still, the performance of these methods is still being judged, is reduced to enough principal components to capture 99% of the variance, which is usually 11 to 20, depending upon the size of the group, and fitting models to those. But that still runs into the problem that you may have more principal components than you can use to estimate parameters. So what we've been using is, recently, is the disparity through time method. I assume many of you are familiar with it. It is not actually disparity through time. It's a proportion of the total disparity within clades. So what it does is partitions disparity at each point along, each branch point along the tree, asking what is the disparity within clades that are present, whose ancestor is present at that time, and then averages the disparity of the subclades and divides that by the total. So what this is a ratio, the relative disparity, and we relabel the axis because it's misleading when it says it's disparity. The total disparity is set to 1. So this is the proportion of the total disparity within clades as opposed to between clades. The MDI is a function of how much simulate Brownian motion, that's the dotted line. And then the confidence interval around Brownian motion, and the proportion or the amount of space between the line for the observed data and Brownian motion is then measured, the area between the curves. Being above the line is positive, below the line is negative. So if the MDI is negative, it means the lineage deviates from Brownian motion below this curve, so subclade. The disparity among the clades is high relative to the disparity within the clades. If it's above the line, like obviously it is for Sciurni, it means the disparity within the subclades is high relative to the disparity among the clades. Early burst is a negative MDI. Ornstein-Uhlenbeck persistent adaptation is positive, above the line, and a late burst can also be above the line. So you see his protozerany deviates from Brownian motion in the direction of an early burst. So that's a model for adaptive radiation. That's protozerany there, and it's doesn't even, I mean, it's below the confidence interval for Brownian motion. These are your familiar tree squirrels, flying squirrels, and chipmunks. Deviate from Brownian motion in the direction of either a single stable adaptive zone or a late burst. And Calosaurinae and Marmotina cannot be distinguished from Brownian motion. This is for shape. We do the same for size. Protozerinae and Marmotina deviate from Brownian motion in the direction of an early burst. So Marmotina, we have an adaptive radiation only along the dimension of size of the ones we measured. Protozerinae in both size and in shape. The familiar New World tree squirrels deviate from Brownian motion in the direction of a single stable adaptive zone or a late burst. Pteromaeinae, flying squirrels, Asians, tree squirrels, and chipmunks can't be distinguished from Brownian motion. So Marmotina, the early burst for size in Marmotina precedes the acceleration of diversification rates. This is one of the groups in which we had a late burst of speciation. And the high speciation rates are maintained on some branches, but rates of shape evolution are highly asymmetric. So on some branches, you'll have high rates, and in the sister lineage, you'll have low ones. And the more disparate groups have low speciation rates and richness. And this is one thing we found in a previous analysis for squirrels, is that rates of evolution of shape and diversification aren't related. Some of our most disparate have the least rich, least rapidly speciating, and the most rapidly speciating are morphologically the most uniform. And prairie dogs and Xerospermophilus have a late burst of both diversification and divergence, but they haven't gotten very far yet. So in a few million years, evolutionary biologists will probably recognize them as an adaptive radiation. So using the analysis now for the recent radiation of tree squirrels into Central and South America, we see shape evolution deviates significantly from Brownian motion in the direction of OU or late burst. This was the same we found when we had other species in the analysis, including European and Asian. And size evolution does not deviate significantly from Brownian motion. So to look at disparity then, Protoxerinae is the most disparate in shape, and that's the lineage that underwent an early burst. Sciurinae and tame tree squirrels and chipmunks are the least disparate in shape, and they fit a model of persistent adaptation in a single adaptive zone. The relatively high disparity of flying squirrels suggests it was a late burst rather than OU, when these two models are typically difficult to distinguish. Disparity should be able to distinguish them. And for size, Marmotana and the African rainforest squirrels both undergo early burst for size. Marmotana is the most disparate. Protoxerinae, however, is less disparate than the flying squirrels, which show a model of Brownian motion. And chipmunks is the least disparate, and it too evolves by Brownian motion. But this is a very young clade, as well as a slowly evolving one. But here we can't just simply equate high disparity or low disparity to any one mode. So summary, based on dynamics of size and shape evolution, there are adaptively and non-adaptively radiating lineages of squirrels. Bursts of diversification are not coupled to bursts of divergence. Bursts, early and late, produce high disparity, and persistent adaptation accounts for low disparity. But Brownian motion can produce both very high and very low disparity. So, to go on to talk about adaptation and convergence, is jaw shape adapted to diet? Well, I mean, it seems obvious, right? What else is it going to be adapted to? Now, size might explain much of the variation in diet, because bite force scales with body size. So species can adapt to eating hard foods by increasing body size. But size might be constrained by other niche dimensions, since giants can't occupy the branch tip niche. And there is one. So there may be limits on how size can be changed. In which case shape will have to be modified to produce higher bite force per given size. So is jaw shape adapted to diet? Documenting adaptation by comparative methods is difficult if convergence is rare, because without convergence, shape won't be predictable across lineages. So what makes convergence unlikely? More than one optimum per ecological class. I think that is common, predictable, because one is the many-to-one mapping of form onto performance. There are many morphologies perform equally well in that many different jaw shapes can produce the same bite force. So if selection is acting on bite force, there are many different jaw shapes, multiple optima. Now there's the one-to-many mapping of form onto ecology. A single functionally versatile morphology can exploit diverse resources. Another is the many-to-many mapping of ecological classes onto function. Ecological classes like shape are multidimensional. Think about diet. Within any diet class, foods can vary along multiple dimensions. So think of fruits. They can be soft and pulpy, fibrous and tough, hard-shelled and resistant to fracture, and Don told me to cut out the rest of my list. They can also be as small as blueberries or as large as coconuts. And squirrels do eat coconuts. There's an idiom in Indonesian, bored through, like a squirrel through a coconut. There's also variation in feeding behaviors that can be used to eat any food of any given material properties, including gouging, prying, shaving, puncturing, and crushing. So therefore, species within the same diet class, as we define them, will have different loading regimes that result in different patterns of internal stresses and strains on the skeleton, and that's what the jaw is adapting to. So here we have convergent diets. The probabilities of most diet transitions are low, but convergent diets are common. The diet that arises most frequently is seed eating, and by that we're excluding nuts. And that has at least 5 independent origins. And there are multiple transitions away from, as well as returns to nuts. And if you're really interested, this is now on early view as of a couple days ago. So what makes convergence more likely? One is niche-dependent limitations on body size in a functionally demanding niche. So they've got to produce high bite force because of the demands of function, but there's a limitation on body size, so they can't just keep getting larger. Then there are going to be fewer jaw morphologies that can produce high bite forces at small size. Additional constraints, such as the inverse relationship between bite force and gait, can further narrow the set of equally optimal morphologies. All adaptations that increase bite force reduce gait. And this is obviously going to be most consequential for animals eating foods that are both hard and large for their body. So taken together, constraints and trade-offs may limit the array of foods that can be eaten and restrict the array of feeding behaviors, producing niche-dependent scaling relationships. So here we looked at size disparity within diet classes. Hard nut eating is the most size-constrained dietary niche in the sense that there's a much narrower range of sizes within that diet class. Bark gouging is size limited, but it's not restricted to miniatures. I mean, you think of bark, if you work with squirrels, you think of bark gouging miniatures, but they're not all actually miniatures. So this shows that there's a relationship between diet and size. And in both cases, we split the lineages because there are different diets represented in the different lineages. So in tree squirrels, diet depends on size, and in ground squirrel, diet also depends on size. What's interesting here is that shape depends upon diet and size and their interaction. So we can't say shape depends on diet, because you can't interpret a main effect when there's an interaction. So what we have is that the full model, size, diet, size, cross diet, explains the variation in jaw shape. C is that we have diet-dependent scaling relationships in tree squirrels. We also do in ground squirrels. As you actually notice, variance explained, it's more impressive in tree squirrels. There's more variance explained by the interaction than by size. Here we have more uniform diets, and we also have the fact that almost every single grazer comes from the same lineage. So to look at ecomorphological convergence, this is a phylogeny. That's a phenogram that's just putting together who looks most similar to whom. So after rotating the two diagrams to maximal similarity in the sequence of species, you can draw lines showing where the position is of species on one diagram to its position on the other. And these are color-coded by diet. Pinkish, the magenta, are the bark gougers. The brownish are the hard endocarp, hard nut eating specialists. So ecomorphological convergence is generally rare. It's most common when body size is limited and diets are challenging. So we have bark gouging miniatures, hard endocarp, that's the hard nut eating. And the hard nut eating protozoarians converge on some silurians that are much smaller. Two other cases might be convergent, depending upon the optimization of diet. We don't know if it's the same origin or different. So here's the statistical analysis. C1 is the degree to which convergence closes up the distance between ancestral lineages. So the p-value of statistical significance of C1 is tested by asking if the degree of convergence in the data is any greater than would be expected for randomly evolving lineages. And so we also, the Procrustes distance there is a measure of the morphological distance, because you can close up a very large morphological distance, 50%, and you're still really different. So we want to know, or not only did they close up, let's say 20%, but how similar did they end up being? So what was more surprising is we also found unanticipated cases of convergence, that these are morphological, without ecological. Now, considering if you're thinking that convergence is going to be rare, and ecomorphological convergence is going to be rare, what's surprising is finding morphological but not ecological convergence. And I'll just say, I can't explain it. So, summary is diet, size, and their interactions explain most of the variation in jaw shape. That interaction documents diet-dependent scaling relationships, and thus that allometry contributes to ecological adaptation. Often allometry is treated as an alternative, intrinsic explanation for ecological adaptation. But niche-dependent scaling relationships are part of the adaptation to the niche. Ecomorphological convergence, however, is rare, being most common in size-limited and functionally demanding niches, bark gouging miniatures. They look like wood chippers. If you look at them, if you see them chewing from behind, you see the wood. I mean, some of these are mouse size, 16 grams. And the wood is being piled out. It's just, they're pretty amazing. The hard nut specialists that you can hear eating even if you can't see them. And the hard nut specialists on smaller bodied nut eaters. So they have the powerful, robust jaw of the nut eaters, but they're also much bigger. To eat what they eat, you need to roast it and chop it up with a machete. So now, coexistence. This map shows species richness. And I say, notice the difference in scale bars. The red on the old world is 28 species. The red in the new world is 12. I love these pictures. I was so thrilled to learn how to make them. And our objective is to do the same, to map disparity, just like we can map diversity. And we're working with Pascal Title on how to do this. And we started putting together the code for calculating all the measures of disparity and figuring out what to do for missing species and cells. Until we can do that, we've quantified disparity in equal area cells of selected regions in North America and the Old World. The graphics aren't going to be pretty. And as well as quantifying variance, we've measured the range of shapes and how similar on average each species is to the one most similar to it, which is the nearest neighbor distance, and whether variation is clustered, uniformly distributed, or over-dispersed. These metrics are commonly used in studies of functional diversity in ecology. And our main aim is to determine whether regional species richness is related to regional disparity. Because one of the questions is whether there's something in environmental carrying capacity and the ability to diversify along multiple axes that explains high species richness. So caveat, we did not take elevation into account. Elevationally zoned regions are therefore treated like one region. I did for some of these, and just going through and looking at which species are present at what elevation and redoing analyses at various different elevations. That gets really difficult when you've got too many areas and too many elevational zones, and you don't have Grinnell to tell you which ones are present where in the past. We also have not measured every species of squirrel, so some are represented by a relative. And we didn't measure disparity for regions that are dominated by missing species, because you can only go so far sticking in relatives, especially when you supposedly have 3 from the same genus in a region. So these are just bar charts. What I've singled out are 3. Ann Arbor, for sort of obvious reasons. but also because it struck me when we finished our first analysis that boreal forests, at least ours, our backyard, had as much disparity as the West African rainforest. What we're counting as regional diversity here is actually one more species than you commonly find in your backyard. 13-liners are not found in most of the backyards that have woodchucks and 2 species of tree squirrels and the little red squirrel and tamias, because 13-liners are moving east, partly because of deforestation and freeways. We find them most often on golf courses, schoolyards. So half a block from us is a schoolyard, and another about half a block other way was a science center. So we could say we lived within half a block of the entire regional fauna. Gabon, that's the African rainforest that was a subject of Louise Emmons' study. So we knew which species actually interacted with which ones and what dimensions they partitioned it on. And Saba, well, because it's extreme and because I got a thing for Borneo. So this shows the variance and this shows the range. One thing you'll notice is that Ann Arbor is close to Saba and above Gabon in disparity, which is a variance. But when it comes to the range, Saba way outclasses Ann Arbor. So first we'll look at this and then I'll show you why. We do find the diversity and disparity, are related across regions. Anyway, here's why we have such disparity, and this is just in lowlands. The 2 extreme morphologies coexist. And some of the other oddities exist. But there are a whole lot of these different species. And the more of the same thing you add, the lower the variance is, because you have many, many similar things and a few different things. So the average of the pairwise distances, or the distances from the mean, goes down. But the range can be huge. So that's what we have. That's only selected. I didn't have room on a slide to put all of them. That's regional diversity and disparity in Ann Arbor. I'm missing one of the species from the backyard. But this is fairly high disparity, but this is the only one that there's another one like. So we don't have a wide range. We also don't have a lot of duplication of the same thing. So Saba and Ann Arbor are similar in variance, even though Saba has a much greater range of shapes, because Saba has many more species and many of them are similar to each other. Those similar species diminish the variance, but not the range. So for nearest neighbor distances, I mean, the 3 places I picked out are sort of boringly similar. Now these are going negative. This is 0 and these are negative. Over dispersion is above the line, below the 0 is clustering. So we see, again, when you have many similar things, you will also have much higher clustering. So for size, Ann Arbor, again, compares to Saba and Gabon. This is because it's the maximum size versus the minimum size. If you've got the same species, you will have the same range. And much of the Midwest, as long as ectodome 13-liners have gotten there, has the same species, plus or minus a flying squirrel, or plus or minus 2. There's commonly 1, sometimes there's 2. And so there's the range. So high there because of miniatures, not because of giants. After all, we've got woodchucks, so we've got giants, but we don't have teeny miniatures. Now the nearest neighbors and clustering, what we're seeing is here, when it comes to size, the nearest neighbors are much more similar. And here, in a sense, what you would expect for size is the over-dispersion, not clustering. If species are partitioning environments by size to maintain size ratios. And we found that in Gabon, species that eat the same foods and are at the same height in the canopy differ by Hutchinson's ratio. So part of what we're interested in looking here is how often do we find that we deviate from Hutchinson's ratio is because they're differing along enough other dimensions, and it's only when size is the one thing that differs do we expect to find Hutchinson's ratio. So summary is boreal forests have very high disparity because of the mix of coexisting lineages and the few highly similar shapes. The range of shapes is far greater in Asian rainforests, primarily because of extreme shapes of some dietary specialists. And the range of sizes is also greater because of miniatures. Shape typically shows a high degree of clustering, but size is more often over-dispersed than clustered. And species richness and disparity are related in our small sample of regions. We don't know whether it's because species-rich communities are highly disparate due to more specialists, more dimensions along which dietary niches are partitioned, or greater regional or elevational turnover among the communities in the species-rich, often montane regions. So, because I've been teased about this for many years, are squirrels really much less disparate than other rodents? This is, again, just for Josh's shape. We've only measured 281 chryseidids so far, and this is why you keep seeing us, because we've measured 186 species of squirrels and 281 species of chryseidids, and we try to get at least 10 specimens per species. So we spend lots of time on these animals. Squirrels are more disparate in shape than chryseidids. Now squirrels, of course, are much older, but squirrels are more disparate. But the range of shapes is greater if we go along all dimensions. Chryseidids have a larger range of shape. But if you just say, what's the most different, the distance between the two most different, squirrels exceed chryseidids. So acknowledgements. I don't put Don on as a collaborator because obviously he's a co-author on the talk. We are really dependent on our collaborators. I don't know if Lucy did make it here, but one of our collaborators is doing a postdoc here. Lucy, are you here? No. She's probably in the lab. Our curators.
She's back there.
Oh. I didn't actually see you, but now I do. Our undergrad research assistants, one of whom is looking for a summer internship. He's really interested in birds. He turned in an abstract and a project that's above what I expect from first-year graduate students. He works incredibly hard. He's incredibly fast. If you have any prospect of wanting a summer intern, and his family's just moved to Berkeley, so he'll be here. Speak to me. He's really amazing. Access to unpublished data, encouragement, support of all kinds, despite disparaging the disparity of squirrels. And the list would go far too long for a slide, Jim. Programming the R development core, without whom none of this would be possible. And as far as I can tell, modern life as we know it would cease. And support from the Museum of Vertebrate Zoology. Thank you.
So I have trying to frame a decent question, but it has to do with diet disparity and your measurement of jaws. Because, I mean, in all rodents, but you think squirrels in particular, I mean, there's two different kinds of bite forces involved in processing food. One is positioning the sizer so they can gnaw, and the other is, of course, positioning the cheek teeth so they can chew.
Actually, it's the grazers I was thinking of because of the medial-lateral translation.
Yeah,
That's
but what I was wondering is, so are there types of food that are restricted to certain squirrels because they can't chew through, they can't gnaw through, you know, the shell like a Brazil nut? If they could, they can get to the pulp, and they can, you know, gnaw it up into small pieces and process it easily.
Incisor bite force is obviously what you need.
Yeah.
For what usually happens is actually they just take longer. I don't know where did this, I had a slide in here that, some of my chipmunk slides I think are gone. I mean, the fact that chipmunks can eat acorns, when acorns are large for a chipmunk.
Yeah.
It's not that they don't, I mean, they don't eat them as efficiently. Some can't eat Brazil nuts, I'm sure. I mean, after all, we can. But most of them can if given enough time. But then there are the grazers. I mean, the grazers really have adaptations for being able to resist torque. Now, the idea that there would be the specializations for cheek teeth and incisors is why we treated seeds and nuts as distinct categories. We thought that the nut eaters would be optimized for incisor bite force, and the seed eaters would be optimized for molar bite force. And then we found, actually, the tanglegram is useful for this. I mean, if you don't mind showing people how wrong we were. The blue and the yellow are nuts and seeds. We see completely interspersed morphologies and whether they're eating nuts or seeds.
And whether you're talking about chipmunks or tree squirrels. Chipmunks at the bottom, tree squirrels at the top. So two very different morphologies also have that same confounding of ability to eat seeds and nuts.
I don't know what happened. It was a wonderful slide, pointing out that there's more to diet than foods. Was that in there? Did I miss it? Chipmunks transport.
Yes, you listed a lot of the different properties, but.
Chipmunks have to transport acorns.
I guess one of the things that was going through my mind was what if you could use your your shape measurements to predict what an individual sciurid can eat or what's beyond its capacity to eat because of differences in bite force at the incisors versus over the molar rows.
As far as I can tell, I mean, you would not predict that chipmunks could eat hard things if you just look at them. And you do have to take into account they eat hard things that are large for chipmunks, not hard things.
Yeah, no, that's.
I
mean, you have to scale the hard object to the body size of the animal.
Yeah. Well, I think a big part of what goes on with chipmunks is food caching, so that they can take it to somewhere they can eat at leisure.
Yeah, they also
And,
eat
you
a
know,
lot of
perfection.
fungi.
Well, everyone does, it's
Yeah.
all that.
Yeah.
I mean, glaucomes,
Yeah, no, particularly
Specialists.
alchemy.
and, I mean, we could see our backyard squirrels eating it, and backyard finding.
Okay.
But to be able to transporting the acorns is a constraint on game.
Yeah.
And the picture that I'm really sorry I don't have in here had chipmunk with 2 acorns, and I mean, and seeds. I mean, it could not fit the second acorn in its mouth. But it's not gonna give up gait to increase bite force.
Yeah.
But it can take the food to where it's safe and then eat it, and it doesn't matter if it's slow, as long as it's protected while it's eating. Yes, Chuck?
You mentioned that in regional disparity, you were comparing most similar pairs.
The nearest neighbors.
Yeah. What would happen to that analysis if you compared closest phylogenetic sister taxa?
I have that. It's phylogenetic nearest neighbors. Let me actually see. This is when it helps to actually have your computer. Bar plots, where is bar plots new? Let's see. I'm trying to figure out. This is phylogenetic diversity. This is actually squirrel diversity by species. That's phylogenetic diversity, and that's the phylogenetic nearest neighbor diversity.
And you get a similar pattern that you got from?
It's different in that part of what I'm not sure nearest neighbors is going to be, well, it's the mean of the nearest neighbors. So like in Ann Arbor, we've got 2 Sayuras, Anatamias Sayuras, but then we've got Glaucomes, Marmota, Tamias, and Ectodermis. So we've got some few that are very close relatives, but a lot that aren't. And Saba here, I mean, here's Ann Arbor's the 4th, so there's Ann Arbor. But one I was actually expecting and was wrong about, I was expecting, to some extent right, that North America would see lower or higher nearest neighbor distances because of all the regions where you have mixes of lineages, where lineages diversified on different continents and came to North America. While in Asia, we do have flying squirrels and Calocyrene. And I think if we look at diurnal communities, we get different nearest neighbor patterns to looking at sort of regional communities, since that will combine nocturnal flying squirrels and diurnal squirrels. But we do want to do that partitioning. The other thing to look at is whether the regional nearest neighbor distances are really that different from the actual community. But figuring out what is a community for rodents is difficult because they're mobile, and you're told if you want to try to get rid of a squirrel from your backyard, take it at least 10 miles away, or else it'll be back pretty soon. And they can walk that, or they don't. Power lines are really useful. Power lines facilitate dispersal in squirrels. I saw a hand there.
So I'm interested in, like, if you take the landmarks on the jaw and you compare across species, whether there are some specific landmarks that are a lot more variable than others. For example, if you put it on like a wireframe grid and try to see where the variation actually sits within these jaws, and whether or not if you looked at like not even just species level but like population level to see how that variation compares to perhaps the species level trends.
Well, I have looked at whether the variance-covariance matrix within species predicts phylogenetic variance-covariance matrix. And part of this is, do patterns of morphological integration, modularity, predict evolutionary integration and modularity? Surprisingly more than I expected. But at this point I've got, what we've done for these analyses is we take 4 photographs of each individual, 2 on the right, 2 on the left. We measure then fluctuating asymmetry and variance and average the 4 replicate measurements, because here we need really precise minimizing measurement error, and also because of Klingenberg's arguments about fluctuating asymmetry, providing information about developmental modularity. So we've got, I guess now, 6 species that we have measured at least 65 to 100 individuals, and we finished digitizing. Now I've only done the comparisons for a couple, and one of the issues is to compare to phylogenetic variance-covariance matrix. What do you do when different lineages evolve by different modes? What's the phylogenetic variance-covariance matrix? For squirrels, when we've got early burst, to get the phylogenetic variance-covariance matrix, you need a model. And would be different models or different lineages. So one possibility is to take variance-covariance matrix, compare it, let's say, take Calocyrene. So Calocyrus notatus is one of the ones we've got variance-covariance matrix from, and from one collecting site. We could do that and then only look at Calocyrene, such as its evolution is. I mean, we've got two species of Tamias so far that we've got. And we could look at evolution in Tamias, such as it is. I mean, I think what we want to do is look more broadly. And also to do the comparisons of covariance matrices. But I'm not convinced, since I've been working on, my dissertation work was on morphological integration. And that's been a main interest before it was called modularity and became a hot topic. It's been one of my main interests throughout my entire career, and I'm not really satisfied with what you get from just looking at matrix correlations. And I'm entirely dissatisfied with what's become the dominant model for mandibular modularity, which is you got the front and you got the back. Jaws don't work like that. I mean, there are two functions, biting and chewing. One is not in the back of the jaw, and the other is not in the front of the jaw. But the idea that this two module came from the idea, one part of the jaw functions to support muscles. The other part of the jaw functions to support teeth. You can tell somebody uninterested in function came up with that as a model.
Well, maybe with that we should thank Miriam for a great talk.